Bismillahirrahmanirrahim. Assalamu alaikum. Welcome to my YouTube channel Science for Everyone. Today in this video I will discuss a very important topic that is related to HIV, and the topic is that why there are no vaccines for HIV. Okay, so this question is asked from students, and different people ask from each other that why there is no vaccines for HIV. So I have searched many articles and videos and even books, and I have asked from my supervisors and my professors, and after that I come to a conclusion, and I find out a very simple answer to this question, that why there are no vaccines for HIV. So stay tuned with the video and watch this video till end, so that your minds are cleared about this question, and you will get the best answer. Okay, so then you can use this answer in your assignments, in your presentations, and you can also give answers to your teachers when they ask you that why there are no vaccines for HIV. Okay, so let's start with the video. First of all, I want to tell you that I will explain it in my way. Okay, so just hear my voice and watch the animation, and I guarantee that you will understand it very well. So don't get bored and just watch this video. Okay, so let's start the video. Okay. Before going to the text, just do not read out this text first. I want to show you a little. I want to present this diagram to you people. After that, I will tell you the main reason that why there are no vaccines for HIV. We all know that our this human immunodeficiency virus consists of a nucleic acid, that is RNA. Okay, the nuclear material that is present inside the HIV is basically single-stranded RNA, and I have discussed in the structure of HIV in the previous videos. But just listen to it, that in HIV there is RNA. Okay, instead of DNA, two strand of RNA. Okay, so it comes in the family of retroviridae, which means that reverse transcriptase is present inside this virus. And what does this reverse transcriptase do? It converts the RNA into a double-stranded DNA. Okay, it converts the RNA into double-stranded DNA. And who performs this function? This function is performed by this reverse transcriptase. And here is the mechanism that RNA is converted into. First, this RNA is used as a template. Okay, it is used as a template, and this reverse transcriptase enzyme uses this strand as a template and synthesize DNA with respect to this template strand. Okay, after this there is no use of this RNA, so the reverse transcriptase have the function that it degrades or it destroy or it completely eliminate this RNA. Okay, so it cuts this RNA into pieces, and this function is also known as RNase. Okay, ribonuclease H. So after that, this enzyme has also the ability of DNA polymerase, DNA dependent DNA polymerase. So then it uses this blue color strand of DNA as a template and synthesize another strand of DNA against it. Okay, so now the single stranded RNA is converted into a double stranded DNA, and after that this DNA is incorporated into the DNA of the host, and it starts its then transcription is done, and translation is done, and different proteins are produced. Okay, and after that these proteins assemble, and a new virus is formed, and then it buds off uh, from the host cell. And this is the general overview. Okay, that first entry of the double RNA strands, and after all this is it is converted into double stranded DNA. Okay, then it is integrated and then it is used, and different messenger RNAs are produced. And after that, some RNA of one strand of RNA is used as a nuclear material. Okay, and the other RNA strand uh, is used as a sequence as a messenger RNA, and is used to synthesize different proteins. And after that, it get in. It is a new virus is formed and it buds off from the host cell. So this was general. This was a general overview. Why I am explaining it? I have explained this pictures in the previous videos, but the main purpose is that I want to make you clear that the main concept that why we cannot synthesize a vaccine. Okay, so now you will come to know that why I have explained this pictures first. Okay, these figures first. Now, come to the text. Reverse transcriptase have no proofreading ability. Okay? Reverse transcriptase have no proofreading ability, due to which it can commit mistakes in DNA synthesis. I have told you that it synthesize complement DNA against the RNA, and after that it again synthesize. It uses the DNA as a template and synthesize another strand against the DNA. Okay, I have discussed it. So who is performing this function? The reverse transcriptase. Okay, it has DNA dependent DNA polymerase activity and also RNA dependent DNA polymerase activity. So these are synthesizing DNA strand across against the RNA template and the DNA template. Okay. So after that, we all know that I want to make you clear that our the human cell have DNA polymerase inside it, and when it is synthesizing, it is replicating when the DNA is replicating by using the DNA polymerase. So our DNA polymerase commit mistakes. Okay, it occasionally places false nucleoside again nucleotides against the nucleotides that are present in the DNA. Okay, the template strand. For example, here is a strand, and there is adenine and thymine and guanine. Okay, for example, it is a template DNA. So what does the DNA polymerase do? It synthesizes, okay, T or also T A, and it occasionally it commit mistake and it place a wrong nucleoside against it. Okay, for example, also T. So this is a mistake. Okay, I want to show you. This is now a mistake. So what does the DNA polymerase do? It then start again it proofreads its strand. Okay, when it proofread it, so it finds the mistake, and then it is cleared by that polymerase. It has the ability. So there is very minute chance of mutation that is present in the, or we can say 0.01% chance of mutation, because our DNA polymerase have uh, proofreading ability. But this ability does not include in the reverse transcriptase. Okay, what does this reverse transcriptase do? It synthesizes DNA strands against the template RNA strand and the DNA strand, and it commit mistakes. Okay, and due to which it have no proofreading ability. Once it commit mistakes, then this mistake is permanent. Okay, because it does not have proofreading ability, and it can lead to mutation, and thus there are random structural changes in the antigenic component of the virus. Once a mutation is caused, after that it does not proofread it, and once it is integrated into the DNA, so that mutation is permanent. And we all know that proteins are synthesized from the genes that are present in the DNA. We all know that. Okay, So if our genes are mutated, so there will be mutated in simple mutated proteins. Okay, then the structure of a protein will be changed. For example, I want to show you that, for example, here is adenine and thymine and guanine, for example. Okay, so uh, I think it could be included in the table that is present in different books. For example, this encodes a protein, an amino acid that is, for example, glycine. It does not, glycine have, the code of glycine is another code, but I'm just telling you, okay, for to make you understand. So when there is a mutation, okay, and this guanine is replaced, or this thymine or this adenine is replaced by an another nucleotide. So this so when it is, for example, this is it, it is now G. So then glycine will not form, and another amino acid will form. For example, that is, we can say lysine. Okay, so I was just saying that when there is mutation, so our proteins, the structure of proteins will be changed. Okay, I was just saying this. Okay, so I think now you have understand it. So when because of this random mutation, there is a random structural change, because the proteins that are formed from that mutation will be in your structure hoga uska, it will be different. Okay, so what will happen? So the species of the HIV virus will not resemble each other. Okay, for example, this is the antigenic structure. Okay, this is the glycoprotein 120. So for example, there is a change in the DNA due to mutation. So there will be, for example, and that the gene that is encoding glycoprotein 120 is slightly mutated. So there will be a minute structural change in the glycoprotein 120. Okay, and I want to tell you guys, just keep this thing in mind, that our adaptive immunity is very much specific for an antigen. Okay, our antibodies that are produced by the plasma cells, the B cells, that they are very much specific for their antigen. So a slight, a minute change in the antigen will cause, uh, will lead to a slight change in the antigen. When there is a slight change in the antigen, so the antibodies will not bind to it. Okay, now then another type of antibody will form against it, that will perfectly match that antigen. Okay, so just because of this random changes in the antigenic structure and antigenic properties of the HIV virus, we cannot prepare an ideal vaccine for the virus. Okay, for the HIV virus. And I want to tell you that what are vaccines. Vaccine are that virus, or the antigenic structure, antigenic proteins that we extract from the virus, and we use it as an antigen, and then antibodies are formed against it in our body when it is injected into our body. Okay, so when this virus is continuously changing its antigenic properties, so we don't know that which, for example, we prepare a vaccine for an HIV, that the glycoprotein have a certain type of structure. Okay, and after that, that vaccine is launched. Okay, so we all know that our this HIV virus is continuously changing the antigenic properties. So again, this virus will change itself. Okay, so that vaccine will no longer perform its function, because now the virus has changed its antigenic property. So we would need a very diverse amount of vaccine. Okay, to treat this HIV, and we cannot do this because we don't know that how much continuously this this changes take place. So there is no ideal vaccine for HIV. and this is just because of the reverse transcriptase. It does not proofread it, and it due to this lack of proofreading, it produces mutational genes. Okay, and when these genes are translated, it produces different structural proteins. And I want to clear to you people that the structural proteins are not that much different from each other, but I have told you a minute change can also cause a change, because our antibodies are too much specific for its antigen. तो अगर हम एक वैक्सीन बना लें तो वो कुछ ही दिनों में वो नॉन फंक्शनल हो जाएगा, क्योंकि हमारे साथ वायरस में कंटिन्यूस चेंज आ रहा है। ठीक है, तो वो फंक्शन को जो इट विल बी उसका जो फंक्शन होगा वो फिर सही तरीके से के साथ नहीं होगा, क्योंकि वायरस जो है उसने तो अपनी एंटीजेनिक प्रॉपर्टीज तब्दील की है। तो एक टाइप की एंटीबॉडी बन गई है, लेकिन अब वायरस ने तो जो उसके सरफेस प्रोटीन है दे आर चेंज, so then our vaccine does not work. Okay, so this was the main reason. I think you have get my answer. If you have any question, you can ask me in the comment box. Okay, so let me know mm, your advices, and if you have any information about this question. Okay, because science is a very diverse field. There are many answers to a single question. So I have found this answer. If you have answers that is related to this question, and when they are authentic, you confirm it from your experts, and after that you share it with me, so I will also make a video over it. Okay, so stay tuned and work hard and educate yourself from wherever it be. Okay, thank you very much. God bless you.